and let me know if I can go introduce myself.
All right, if you could please find a seat.
Right, so she knows who
We'd like to start on time. So, welcome everyone to the 2014 CF Education Day. It's a great day for us. It's when we come together, both from a center team and a CF community, to sort of catch up with each other when it comes to where we're going with CF, what are the new advances, and where we think we're heading over when it comes to new developments and trying to improve on every aspect of what we do for CF patients and their families. And I have to give a few sort of housekeeping announcements. First of all, and this is very important for us, as you probably are well aware, infections are an important piece of the whole CF pathogenesis, so we're very conscious about infection control. So we are following very strict infection control precautions. We've always done that. We really haven't changed much in that sense. So please use your hand gel generously. We have plenty of stuff to give away. Use it frequently. And for those who have CF, we are providing you with a mask. And please try to stay at least 6 feet away from each other. And also use your hand gel generously. And thank you for being here. For us, it's very important to have you here. Secondly, I'd like to obviously thank again to CFRI for their tremendous support and particularly for helping us have a web presence. We know many of you come from really far away as patients that are care at the Stanford Center and also as participants of our multiple research studies. So we like to do our best at having you participate of this meeting, and having a web presence is a major way of doing it. So thanks for CFRI for helping us stream live. So now we can say live from Palo Alto. The 2014 Stanford. So thank you. Now this room is going to be the main room for the morning sessions. In the afternoon, we're going to break, as we've done on previous years, into pediatric focus and adult focus sessions. The pediatric sessions are going to be on this room, the adult sessions on the room across the hall. And I'll ask you to please, when we break for lunch, take all your stuff away because we need to reorganize the chairs, so don't leave anything on the room. Now, to get officially started, I'd also like to ask you to please fill your evaluations. They are in your booklets. It's very important for us to hear your opinion about how the day went, what was important, what worked, what didn't work, and particularly what would you like to see on future conferences. Because believe it or not, Colleen, who is probably doing something right now, she's very busy putting this together, and we really need to give her a big hand for all the work that she does in putting this conference together. We usually start just the Monday after the conference thinking about the following year conference. So it takes quite a bit of work to get this put together. So thank you, Colleen, for all of your effort doing this. And in great part, what helped us think about what to do for the following one is all your feedback and input as to what would you like to see. So without further ado, I'd like to introduce our featured speaker this year. It's a great privilege to introduce to you Scott Donaldson from the University of North Carolina, where he's the adult center director at UNC. It's a fairly large adult program and also a fairly large CF program with well-established reputation in advancing the science of CF, which has helped us tremendously in being able to think about where to go with not just care, but also new treatments. So no one better than him to give us a review as to where we are in understanding CF and how we're using that knowledge to develop the new therapies for CF. So Scott, if it is okay.
Thank you very much. It's really an honor for me to be here today. I was really excited to get the invitation, and really appreciate all the work that Carlos and Colleen especially has done to organize this session. You guys really have a special CF center here. You probably realize that already, but I think it's worth mentioning. The people that are taking care of the patients and doing the research are so devoted to this purpose in life. And last night at dinner, kind of going around the table, recognizing so many people have been doing this for 20 and 30 years, really devoting their entire professional lives to taking care of CF and moving the research forward. So it's a special place. I hope you appreciate it. It's really, I think, exciting, too, to see so many people with CF and families of people with CF who take the time on a Saturday to drive here. And I understand that's not an easy thing frequently in California. That, you know, this is important to you too, and you're invested and you're really part of the CF team here as well. So it's exciting. It's to work together with you, and to you know, it gets me energized to go home and work in CF as well, having so many people that care about this. So anyway, thank you. Carlos said it perfectly. You know, my goal today is to really talk about some of the research that's happening recently and how that's driving us toward better therapies for CF. And this is really a special time because of these recent advances that we've made. So this is sort of the outline of what I want to do kind of a quick look backwards to see how we got to where we are today, looking at the science of understanding cystic fibrosis and actually CFTR, which is now really our key target for therapies, and then how this is bringing us forward to trying to fix CFTR, the basis of this disease. And so in thinking about what we understand about CF, and particularly CF lung disease, it actually goes back a long time. And if we go back to even the 1940s, you know, this was a time when, unfortunately, babies born with CF oftentimes did die very early in life, even the first year of life, not because of their lungs necessarily, because of gastrointestinal disease. So obviously that's a tragedy. It's a horrible thing. We're glad that doesn't happen like it did in the past, nearly as much. But it did give us an opportunity to begin to understand the lung disease. So these babies dying very early in life from a gut problem, we had the opportunity to look at their lungs and give us a glimpse at what are the earliest changes in the CF lung, because that's really an important key to understanding the lung disease. And what was observed is that, first of all, these babies likely had normal lungs at birth, and the really earliest lesion that you could see in their lungs was this very bland mucus plugging. And for those of you not used to looking at lungs, this is an airway that's kind of cut on its side. This is an airway kind of cut in half. And what we see inside that airway is mucus. So we think the earliest lesion is just mucus obstructing airways without infection, without inflammation, and that those things follow on later. So even 60 years ago, we knew this is a mucus problem, but we didn't yet know why we had this mucus problem. And so if we fast forward a few decades, some of the really important people in CF who are still working hard at CF gave us really important insights into why we might have a mucus problem. And it actually centers around abnormalities in the salt and water transport across the epithelia that line lots of our organs. And so this is actually demonstrated in living, breathing people. So these are studies called nasal potential difference studies. You may have heard of them. It's a way of actually measuring ion transport activities in the nose. And what we saw, again, going back more than 30 years, is that there was a clear difference between people without CF and people with CF, where we saw really a large increase in the electrical activity in the nose. And this reflected having too much sodium transport. And then by changing the conditions but doing the same type of experiment, we saw a second problem. So there's too much sodium transport, but also in response to specific triggers that should make a normal person secrete chloride, we saw none of that in CF. So we saw there's two problems: no chloride transport and too much sodium transport. And really working about the same time, but now working in sweat glands from people, we saw Paul Quinton coming to exactly the same conclusions, that really that key ion transport abnormality in CF was the inability to secrete chloride. So now we had a view. So we have a mucus problem, and it probably relates to having abnormal salt and water transport that makes that mucus too thick and sticky. And then just a little while later, in 1989, we got a huge breakthrough, and that's actually discovering the CF gene called CFTR. And so this was really a multi-investigator effort. People are really coming together, no longer being competitors, but actually being teammates to make this happen quickly. And this was a huge breakthrough that really gave us, first of all, a very perfect understanding that CF is caused by an abnormal chloride channel, and that's what CFTR is, but also gave us an important tool to then move down the road to figure out really the details of this disease. So just to back up for 1 second, this is really our understanding of CF lung disease. We have an abnormal gene that leads to an abnormal protein that causes abnormalities in salt and water transport. This makes mucus abnormal so that it doesn't move out of these organs well, and that ultimately leads to chronic lung disease with infection and inflammation. So, on the surface, that discovery in 1989 of finding the gene made a lot of us very excited that, gosh, in 5 years we'll have this licked. We'll do gene therapy, we'll put a normal gene back in. But unfortunately, that turns out to be a really difficult thing to do. But having knowledge of that gene and that protein, it set the stage for, instead of just fixing it very easily like we thought we might, but to really have a deep understanding of this protein and what goes wrong. So I want to describe a little bit about what we've learned since discovering the gene. So this is a little cartoon of the CFTR molecule, and you can see that it's a complicated gene that makes a pretty complicated protein with lots of different domains. Each has its own special functions. And if that weren't complicated enough, all of these special domains have to be folded exactly right. So this little loop has to be next to this little loop. And if you get any of this wrong, things fall apart and it doesn't work. So this is a complicated molecule. And it's incredible that the cell can even do this. And sometimes it gets it wrong and it gets rid of anything that's slightly abnormal. But this is also the challenge in front of us. We got to take something that's abnormal and now make it normal. And thinking about this, and when people proposed to try to do this, it wasn't clear that it would work. But today we're going to find out that likely it is working. So that's a little bit about CFTR. But what is it really doing? So it's a complicated molecule, but what are its key essential functions in human beings? We know that in CF there are a lot of different disease manifestations, from the sinuses and nose to the lungs to the liver to the gastrointestinal tract, to the male reproductive tract. So it's complicated disease. But I think it's useful to kind of pick out really what the key functions are. And by focusing on these key functions, we know what we need to fix, but we also know what we want to measure when we assess new therapies directed at CFTR. And so I'll propose to you today that I think the key functions are, first of all, pancreatic function, intestinal function, and lung function. And so we'll use that to pick out what we want to measure. So let's start with the CF pancreas. And cystic fibrosis actually got its name from the pancreatic lesions, these cysts, and this fibrosis that's present in the pancreas. And what is CFTR doing there? So these are very special micrographs at very high power in the CF pancreas, and we can see that essentially there's two portions of this gland. There are these cells called the acinus in the cartoon over here that are responsible for making digestive enzymes, and then there are ducts that attach to them that are responsible for moving those enzymes out of the pancreas to the intestinal tract where they digest our food. And so where we look, where exactly is CFTR in the pancreas, we can see that in this bright green, it's lining the inside surface of those little ducts. So that's where CFTR is. And so what is it doing there? Well, we actually know that CFTR is not only a chloride channel, but it's also a bicarbonate channel. And in the pancreas, it's responsible for making more than 1/2 gallon a day of this bicarbonate-rich fluid. So that's what CFTR is normally doing. And why is that important? Well, in the normal pancreas, we know that this abundant bicarbonate-rich fluid is necessary to essentially flush out these pancreatic enzymes and get them where they need to go, inside the intestine. And what happens in CF? We lose that capacity to secrete bicarbonate. As a result, we actually wind up with these mucus plugs inside pancreatic ducts. And these plugs block the pathway, and so these enzymes can't get out and get to the intestine. And so instead of digesting our dinner, these enzymes wind up digesting the pancreas itself. And this is why very early in life, the pancreas is typically destroyed in people with cystic fibrosis. And so to kind of wind that up for the pancreas, we know that CFTR is really critical for bicarbonate secretion in those ducts. But what does this mean about if we can now turn CFTR function back on? What do we expect to happen in this organ system? Well, first of all, we're quite sure that to really have a full effect, we're going to have to do this very early if it's going to be possible at all. The pancreas is destroyed early in life, and so we're going to have to take action very early on to preserve any of its exocrine function. But on the other hand, there's about 10% of patients with CF who have preserved pancreatic function. So they have milder mutations that retain some pancreatic function. So this may be very helpful for them to, first of all, preserve that pancreas, but also perhaps to prevent these bouts of pancreatitis and inflammation that the patients have. But you know, even in the setting of a pancreas that's been destroyed to a large degree, I think there may also be benefits just by restoring that bicarbonate secretion that occur downstream in the intestine. So restoring that bicarbonate secretion may have other good effects in patients in terms of their digestion. Usually when we think about digestive disease in CF, we're very focused on the pancreas, but we're, I think, learning more and more that CFTR in the intestine is incredibly important as well. And once again, in the intestine, CFTR's job is both as a chloride channel, but probably very importantly as a bicarbonate channel. We know this is expressed to a large degree throughout the intestine, particularly in the early parts of the intestine after the stomach. And you know, some of the really important insights about CFTR function in the gut actually comes from the CF mouse. So the CF mouse was really the first animal model of cystic fibrosis that was made, and the hope is we would have a great model of lung disease. Well, it turns out that the CF mouse has very bad intestinal disease, but almost no lung disease, and in fact no pancreatic disease. So although it wasn't what we were after when we made the animal, it gives us a beautiful model of what happens in the intestine. So it's a pretty pure model of intestinal disease. And what we can see in these mouse, when we compare a normal mouse to one of the CF mouses, labeled in green here is mucus. And we see that mucus is really building up between these crypts that are a part of the intestinal tract and apparently becoming stuck to these surfaces. In fact, when we subject these tissues to conditions that normally wipes away and washes away mucus from a normal intestinal surface, we see mucus, I don't know if that projects very well, but we see mucus that stays stuck to the CF intestinal lining. So the mucus is not behaving normally, clearly. And we're learning and that travels down your intestine and takes pictures of the gut along the way. And we actually see abnormalities that are visually apparent. So we think this is probably a very important part of what's happening in the CF intestine. Let's skip through that a little bit. And then what about the lung? These are beautiful work done by Jeff Wine, who I just saw out of the corner of my eye over here, as well as folks in Iowa working with a CF pig, another very useful model of CF disease. And we can see that clearly mucus is important. The mucus is abnormal. This is mucus being pulled out of airways from a patient at the time of transplant and from a CF pig, and it just doesn't behave normal. It's like rubber blocking those airways. And these lower pictures, we see mucus in the CF pig that's layered on top of the airway surfaces, and ultimately that leads to what we see in the bottom right of mucus filled with infection, filled with inflammation. And we've talked a bit about this, but just to re-emphasize, how does abnormal salt and water transport lead to lung disease? Well, in the normal lungs, we see again a very high power view of an airway cell. CFTR is at the surface of that cell, and it's responsible for making sure there's just the right amount of fluid that's secreted into that airway space so that we have enough fluid for cilia to stand up and beat and move things out of the lung. And that keeps the airway nice and open and clean. In CF, with the absence of CFTR, we don't have enough of that fluid secretion, and so this layer of fluid becomes very shallow. You actually see the cilia that are kind of bent over on their side, so they're poking out of the picture, looking straight at you instead of standing up and down, and the mucus becomes thick and dehydrated and sticky, and this causes the airways to become obstructed and sets it up for downstream infection and inflammation. But in fact, it might not just be about mucus clearance. So CFTR may have other roles in defending the lung as well. And this is work done by the group in Iowa using the CF pig once again. And what they've observed is that when they take airways from these CF animals and they expose them to bacteria in a very special way, they don't, normal airways are able to kill bacteria. And we can see that in blue as a non-CF animal, and that within minutes, most of the bacteria are killed just by the native molecules that are secreted by the airways. The CF animals, on the other hand, do a much less good job of killing those bacteria. So there's a large difference between the normal and the CF animals with fewer killed bacteria. So this is not related to clearance whatsoever, but it's just an intrinsic ability to kill bacteria. And so what is all this about? Well, I've been talking a lot about bicarbonate, which is one of the things that control the acidity in your airways, or pH. We can see that indeed in these CF pigs, the airways are more acidic, so the pH is lower. And pursuing the hypothesis that perhaps this is responsible for the abnormal killing, what these investigators did is took a CF airway, they exposed it to some extra bicarbonate through an aerosol and showed that this is able to increase the pH, but importantly, it was also able to restore a lot of that killing of bacteria. So this is another piece of evidence that CFTR has an important role not only in clearing the lung of mucus, but it may also have an important role in preventing those early infections that children are exposed to. So the question is, how can we fix it? We are understanding what CFTR is doing. How do we fix the problem? Well, once again, the story gets complicated because CFTR is a gene that doesn't just have one mutation, but it has almost 2,000 different mutations within it. Fortunately, these many mutations can be grouped into sort of flavors. So we have class 1, 2, 3, 4, 5, depending on the consequence of the protein. So there are some mutations that make little or no protein at all, and that's called a class 1. We have class 2 mutations, we'll call them maturation, where the channel is made, but it's misfolded, it's shaped funny, and so it's recognized as abnormal, and it doesn't make it up to the surface of the cell. And then we have a group of mutations where the protein actually does get all the way up to the surface of the cell, but it doesn't function normally. So either it doesn't open and close, which is what an ion channel should do, or chloride doesn't pass through it, it doesn't regulate normally, or there's just very little of it there. And so I'm going to simplify this a little bit, and we'll just talk about conceptually that there are mutations where there's no CFTR at the apical membrane, and that there are those mutations where the protein makes it up to the surface of the cell, but it doesn't function normally. And so in thinking about treating CFTR problems, there's essentially two approaches that get at these two problems. So we have the approach that uses a potentiator, which would be a molecule that addresses mutations that make it to the surface of the cell, but it doesn't function. So these molecules will make an inactive or quiet ion channel and make it an active ion channel doing its job. And so the classic or key example of this is VX770 or ivacaftor, which has now been approved for a small subset of patients with this type of mutation. The second approach of treating CFTR directly is with correctors. So correctors are a class of compounds that are designed to move CFTR that gets stuck inside the cell up to the surface of the cell. And so the key examples of this would be VX809 or VX661, which are two molecules currently in clinical trials in patients with CF with a common delta F508 mutation. And this is why CF is very exciting right now. This is treating the heart of the disease, and we have data that when we do this successfully, when we can treat CFTR successfully, we have profound, dramatic improvements in the patients. And so this is the data that was published now, going back 3 years almost, where we studied the drug ivacaftor in patients with one specific mutation, that G551D mutation, which is a mutation at the surface that doesn't open and close. And what we saw in these patients is their sweat chloride became almost normal, but not completely, and their lung function in a very short period of time dramatically improved. And so this was incredibly important proof of concept that if we fix CFTR, we're going to really help patients. Not only did their FEV1 or lung function get better, but the patients were better in terms of how often they got sick. So they're sick much less often, and in this slide shown, they weren't put in the hospital nearly as much. So this is, again, incredibly important that fixing the CFTR is going to be what really is going to make the difference. And so when ivacaftor was approved, I know you probably all had the same thoughts, and my patients were asking me, well, what about me? I don't have G551D. Could this drug help me? Are there other drugs to help me? And so we're now, in the last couple of years in particular, really moving toward the capability and the prospect of actually helping many other patients with CFTR with different mutations. And so I think one way of starting to look at this is there anybody else other than patients with G551D that we can help with this drug ivacaftor? And so the obvious question that everybody asked at first is, what about delta F508? I've sort of described that this is a mutation with a really different problem than G551D. We didn't really think it would work, but it was worth a shot. We did the trial, and sure enough, as we expected, by itself, ivacaftor doesn't help people with the common delta F508 mutation. But what about patients who are even younger than the ones who made it into the clinical trials initially, so under the age of 6? Is this drug going to help those patients? And we don't formally know the answer to this. I think we all believe that's the right approach. And this is formally being tested in a clinical trial called the Kiwi study in 2 to 5-year-olds and patients that have G551D and other mutations that are very similar to it. So we expect to have those results relatively soon. What about the other end of the spectrum? People who are very, very sick, too sick to be in the clinical trials of this drug, who may even be waiting for a lung transplant, for example. Is this drug useful in people that sick? And the good news is it doesn't look like you're ever really too far along to get benefit from fixing CFTR. There's actually been 2 publications in the last year where this drug was tested specifically in people with G551D who had really severe lung disease. And in both instances, we saw very clear benefits in these patients, improved lung function and reduced frequency of exacerbations. What about other mutations? In particular, mutations that are similar to G551D that are what we call gating mutations, ones that don't open and close properly. Will it be helpful for them? And so there's, just for background, there's 8 or 9 other mutations that would fall into this grouping. And fairly recently, we actually have the answer to this via the connection study where it worked. And if you have a handout, the slide said pending approval. It was just about a week ago, actually, that the FDA ruled that this works. It's approved, and now we can start treating additional patients with this drug. And we expect to have exactly as much benefit in these patients as the patients with G551D. So that's very exciting. What about mutations that are different but are at the surface of the cell as well? The classic example of this is the R117H mutation. So this is a class 4 mutation. These patients have some residual CFTR function and tend to be a little less sick, but still can have significant disease. What about them? Well, we very recently completed this trial as well. It's called the CONDUCT study. And the question is a little less clear. And my interpretation of it is that as the sicker you were, the more benefit you got. So adults, people who had more substantial, recognizable disease benefited. People who were very mild, who had with normal lung function essentially, who were very young, it wasn't as easy to show benefit in those patients. So this is going to be up in the air a little bit, and we'll have to wait and see a little bit whether the FDA is going to approve ivacaftor for these patients as well. But clearly, this is all great, exciting news, but clearly Delta F508 is our target. And we have, we have a drug now for these patients. They account for about 6.5–7% of patients with CF. But we know that half of the patients with CF have 2 copies of the common Delta F508 mutation, and another 40% have 1 copy of the Delta F508 mutation. So if we have a great therapy for that mutation, we may be able to help an additional 88% of the patients, unfortunately leaving still about 10% of patients who have a different set of problems, and there's work being done to help them as well. It's just not as far along. So let's think about these correctors, this VX809 type of molecule, which has been given the name lumacaftor. Once again, as a reminder, what this drug is doing is moving CFTR up to the surface of the cell. You see a picture of it here where CFTR is inside the inside of the cell in green here, but after treatment, it moves up to be exactly where it should be, at the apical surface of these epithelial cells. But we know, unfortunately, and have done the trial, that this drug by itself isn't enough to make patients better. Okay? And the reason this is because the Delta F508 channel, it has 2 problems. So one problem is it doesn't get to where it needs to be, but it has a second problem. It's not a very good chloride channel. And so the concept that we're pursuing now is that these folks actually need 2 drugs. So they need a drug, VX809 or similar drugs, to move it to the surface of the cell. And they need a second drug, specifically ivacaftor, to turn it on. And so that's the direction of research right now. And this is some studies done in cells from patients with CF, and it kind of demonstrates this concept. So an untreated set of cells have very little CFTR activity. If you treat that with the VX809 or lumacaftor alone, you get some restoration of activity, but not enough. But then when we come with the combination treatment approach, we get more activity. And the hope is that this is going to be enough to have clinical benefit. And so this approach was launched into phase 2 trials now a couple years ago in patients with the common mutation Delta F508. And so the first study that was done using VX809 and ivacaftor had a design where first patients took the VX809 by itself alone for 4 weeks, and then the second drug was added in for a second 4 weeks. And what was observed during that first 4-week period when they were on 809 alone was that patients certainly had no benefit. Their lung function actually tended to slide a little bit, and even a little bit more than the placebo group for unclear reasons. However, when we came in with the second drug, the ivacaftor, on top of it, very clearly those patients now getting combination therapy had improvement in their lung function, about an absolute increase in about 6%, so pretty healthy improvement during that second 4-week block, whereas patients getting placebo had no further change. So that's pretty encouraging. As sort of a second layer of evidence that this really is working and is really the right approach, comes from a different phase 2 trial with a different drug, VX661, that's similar to VX809, but has been tweaked to have some different properties. And so data from this trial showed that, first of all, when you treated patients with placebo, nothing happened. as you would expect. But then when we came in with a low dose of drug, we actually start to see improvement in lung function during the combination treatment period, that when we took drug away, went back down to baseline, being a good line of evidence that this is really a specific effect. And then when we came in with the higher doses of drug, we had an even larger improvement in lung function, again, being in the order of about a 5% absolute increase or a 8% relative increase in lung function. And again, when you take the drugs away, the lung function falls back down to the baseline. So really, again, really helpful supportive evidence that this is the right approach and it's beginning to work. So this was actually enough data to make us excited and to move forward with phase 3 trials, which are the definitive trials that will decide whether a drug will get approved. And so to do this efficiently, these trials actually were planned and 2 were done at the same time, just to cut the amount of time that's needed to reach approval. And so really gratifyingly, and a tribute to the people working in CF and the patients who want to get these drugs tested, 1,000 patients were entered into these 2 studies in an incredibly short amount of time. And they've been fully filled with patients. And now we are beginning to reach the end of the 6-month or 24-week blinded period of treatment, and patients are currently rolling over into open label treatment where they're getting drug, we know, and to be followed for another 2 years. And so the key endpoints that will be reported on are the lung function of these patients, the FEV1, but also paying very close attention to their nutritional status, exacerbation frequency, and of course safety. And so, as I said, the studies have been filled, and we're actually expecting results from these studies this year, middle of the year. So we'll be holding our breath until summer to see how these turn out. And so, we're excited, no doubt about it. But I don't think any of us are satisfied with where we are today. We know we have to still do better. Okay, we want to make these drugs better. We want the kind of activity that we saw with ivacaftor and G551D or more, because we think that's going to be necessary to get the maximum benefit from these drugs in patients. And so, to continue to move forward, we need better drugs, but we also need better tools to test the drugs, and that's what I'm going to talk about a little bit now. So first of all, how do we get better drugs? Well, to do that, we have to understand how the current drugs are working. So I'm going back to the little model of CFTR here. So we have the different kind of key domains in the molecule. And as I said earlier, it's important how things are folded and how they associate with each other. And one of the key associations is between this NBD1 domain and this MST, it doesn't matter what these letters mean, and this MSD2 domain. So they have to be close to each other in just the right way for things to fold properly and to work. And we know that with the delta F508 mutation, which is located in this NBD1 domain, that interferes with that association. And so this little cleft isn't right. And so that is a huge problem. There are other problems as well, though, with that delta F508 mutation. It actually makes this NBD1 domain a little bit unstable. So it's kind of, instead of being a nice firm ball, it's kind of a wobbly ball. And there are further downstream problems of making the second NBD2 unstable as well. And so we have multiple problems that we have to correct. The drugs that I've mentioned already, VX809 and VX661, we're now learning exactly how they do work, and they appear to be really addressing that key interface between the NBD and the MSDs. Okay, so that's where they're working. But what if we were able to actually go in and start fixing the other problems, the NBD2 stability, the NBD1 instability? And we have chemicals in the lab that can do that. And so what happens when we start mixing these together to try to fix all of the problems? Well, this is a graph that kind of demonstrates this. So here we have the effect of just using the VX809 on the molecule in the lab. It's an effect of that about that big. But if we add 2 drugs together from different classes, we get a much larger effect. And if we add 3 drugs from the 3 different approaches, we get even greater impact on restoring CFTR function. So this is not only just adding things together, it's actually multiplying effects. And so we're very hopeful that when we can go from having chemicals in the lab to having medicines that do the same things, we're going to get much larger effects in patients as well. So it's very exciting. But how can we then rapidly take drugs that are being searched for and then test them so that we don't have to do trials that take 2 years to finish and take 1,000 patients? Can we have some assays that can more rapidly test individual drugs and figure out which ones are the best? And so one of the really exciting assays that's been reported in the last year are these little intestinal organoids. And these are little structures that are obtained by, first of all, getting a tiny piece of rectal tissue. Sounds like a scary thing, but in fact it's an incredibly benign, safe, painless thing to do. It takes a couple minutes, so just a little pinch of tissue. It's then put in a very special culture conditions where these few cells actually can multiply, and then they grow up into these little balls, and inside these balls they express CFTR just where it should be, at the inside, the inside surface of these spheres. Then we can actually test the function of CFTR using them. So we go from having a little piece of tissue to having lots and lots of these organoids that we can do lots of experiments with. So what do we see? So here's some organoids from a patient without cystic fibrosis. We can see that we have all these kind of funny shaped little balls. And when we stimulate them with a drug that turns CFTR on, I think you can see them growing very nicely. And so we can quantitate how they grow to reflect on CFTR function. What about in patients with CF doing exactly the same thing? Well, not much happens. And so that's the problem. No CFTR function. This gives us a very nice readout of whether CFTR is working or not. And in fact, this technique is actually able to discriminate between not only a healthy person in CF, but also between mild CF that has a little bit of CFTR function and more classic severe CF. And importantly, anytime you start culturing cells, things can change, and we can go from reality to fiction fairly quickly. But in this case, it looks like this actually is really reflective of what's happening in vivo, that if we compare the organoid answer to CFTR function to what we measure in a freshly excised piece of tissue, we're getting the same result across severe CF, mild CF, and control. So it looks like it's there's a high fidelity in the system to giving us the answers we want. And does it report on the drugs that we're interested in using in fixing CFTR? And this is the kind of data we're getting. So taking the organoids, treating it with one drug, individually, or combinations of drug. And what do we see? Again, we're seeing a certain amount of activity with one drug, VX-809. We're seeing much larger effects by adding 2 drugs together, and even more effect by adding 3 drugs from different classes together. So that's, again, incredibly exciting news that we may have synergistic effects by getting drugs from different classes. And we know this is all about CFTR, because if we then add an inhibitor of CFTR, it takes all of that swelling function away. So it's real. So this tool is going to be great for testing drugs, but also gives us the possibility of doing what we call personalized medicine. As I mentioned before, Delta F508 accounts for a lot of patients. The mutations that we screen for and our kind of typical screens for mutations accounts for a lot of numbers of patients, but there are so many other mutations that it doesn't cover. So if we look at the most common 23 mutations, that covers about 85% of patients, but only covers about 1.2% of the actual mutations. So there are going to be a lot of patients out there with unique mutations that we don't know, that we can't do a clinical trial on. So we need a tool to tell that patient if drugs that become available are going to work for them. And so this gives us the ability to do that. And so this is another reason that this is going to be very useful. But this type of tool is great, but we also want to be able to have tools that study CFTR function in living, breathing human beings as well. And the study that was recently completed, called the G551D observational study, or GOAL study, was designed to do exactly that, to look for new tools for studying CFTR function. And so this was designed and set up by folks at the Cystic Fibrosis Foundation with a hypothesis that if we're now in a situation where we have a drug that's going to turn on CFTR in patients with G551D, if we can study those patients, collect samples, do novel novel outcome measure studies, we're going to learn what it looks like to turn CFTR on, and that's going to give us a tool to then study new drugs. And so what did we do? We've talked about what the important functions of CFTR, and in this study we selected a few to study specifically. One is using a method of actually measuring intestinal pH, because as we said, the pancreas and intestine, the key function of CFTR in those organs is to secrete bicarbonate and therefore increase pH. We did a study to measure mucociliary clearance in the lung. And then we also looked at the effect of turning CFTR on infection in CF, which we know is an incredibly important part of the disease. So this was the overall design of the study. It was more than 100 patients got enrolled into this with that rare mutation. We saw patients repeatedly over time, collected lots of samples, lots of sort of standard clinical data, but then we had these sub-studies that were embedded within it. And I'm going to tell you about a couple of them, the intestinal pH study and the mucociliary clearance study. So how, pray tell, do you measure pH in somebody's intestine? Well, it turns out that there's another kind of cute device that can be used called the pH pill. So once again, this is technology being built in a tiny pill about the size of a dime. It gets swallowed, and it actually transmits data from inside of you to a little recorder, to your little Bluetooth recorder that you wear on your belt. And so it tells us the pH inside of you, the motility of your intestine, the temperature, etc. And so the hypothesis was that if the intestinal pH is indeed low in CF, turning on CFTR should fix that. And so the first study that was done was actually just comparing people with CF from people without CF, and we can see that indeed this technique was able to show a large difference in intestinal pH in people with CF and here, where the pH was very low, so very acidic, versus people without CF that had a more neutral pH. This is probably very important because for your pancreatic enzymes to work, whether it be ones you secrete or ones that you take in a pill form, you need a pH in this at least 5.5 to 6 range. So they're not going to work until your pH gets up to that level. So you can see in the CF patients, this is a problem. Your pH is substantially below that for a long period of time. So what happened in the G551D patients who began taking ivacaftor? We can see that before the drug, they certainly had the same pH problem as what I just showed you, but after starting to take ivacaftor, they had normalization of the pH. So the pH now rapidly comes up into the desired range and stays up throughout the transit in the intestine. And so what are the implications of this? Well, first of all, as I mentioned, I think this may be very important to make the enzymes taken via pill form by patients work better. I think it's also going to be very important and likely is responsible for why patients felt so much better. They gained weight, they had fewer gastrointestinal symptoms, and did better from that GI standpoint. So fixing the pH problem is likely doing very good things for these patients. And ultimately, though, doing this as early as possible is more likely to have the most benefit. So what about the mucociliary clearance study? So again, the hypothesis of this study is that if we are able to flip on CFTR, like turning on a switch, that should restore mucus clearance, if indeed CFTR is an important part of that pathway. So in this study, we were able to recruit 22 patients into it. The studies were actually done at 4 different CF centers across the country, and we made measurements at baseline, a month after starting to take the drug, and 3 months after starting to take the drug. Let's get through this. And the open question, I think, was still that, is CFTR really responsible for mucus clearance, or is the abnormal mucus clearance that's been reported just because you have bad lung disease? And this is a study to also answer that question. So this is how we measure mucociliary clearance. I won't get into too many details, but essentially we take a low-resolution X-ray to know exactly where the lungs are, where the outlines are. Patients then inhale an aerosol that contains radioactive particles, and this is a very low-energy radioactive particle. They then sit in front of a camera called a gamma camera, and that takes pictures such as this one here that shows where those radioactive particles are. And then by taking pictures, serial pictures over time, we can measure how quickly they're moving out of the lung. And so this is a movie that's been made by just putting these serial pictures together. And to orient you a little bit, we have the outline of the lung on the left and right here. This bright spot at the bottom is the stomach. And so anything that gets swallowed goes there, of course. And in the middle, between the lungs, is a trachea, so you may see some particles moving up in that direction. And let's first see what these movies look like before starting ivacaftor. There we go. And so yeah, you get the sense that the lungs are getting a little less bright. We're getting some accumulation of tracer in the stomach. You saw some images where things were up in the mouth, actually, from moving up. And so not real exciting, but things are moving, but moving slowly. What about after taking ivacaftor for 3 months? We can see something now, right? So this is that was fun to see. And this is obvious. I'm going to not show you the worst movie we ever had, but this is probably the best movie we ever had. But it's very clear. We do a lot of these studies, and we sit in front of the camera and look at pictures, and it's not very often where you can just sit there and go, this is working. And that's what we see with this drug. This is working. Oops, sorry. I'm not showing off. I didn't mean to do it twice. Sorry. So this is the summary data of what we saw in the whole group of 22 patients. So the black line is how fast things cleared out before treatment. The blue and red line is how fast things cleared out 1 month and 3 months after starting the therapy. And frankly, we've tested a lot of drugs, and we've never seen anything quite this effective. Okay, and so this is a tremendously large impact on mucociliary clearance. And so, is this going to help patients? Well, we certainly think so. We think it's probably related to getting mucus out of the lung and therefore improving lung function. But what might it do downstream from that? We want to not only get rid of mucus, we'd like to help infection and inflammation as well. And I think the surprising data that came from the overall goal study is, in fact, we see evidence that the frequency of seeing Pseudomonas in patients actually decreases once we started this therapy. So there are probably patients who are coming and going with Pseudomonas that might not have a super firm foothold in the lung, and by starting this therapy, we can actually clear it, so the patients can clear it. And so, again, this is incredibly exciting data that by restoring CFTR function, we may have benefit on infection as well. And so, I hope I've shown you data that you find encouraging, exciting. We certainly feel like it is, but we want to keep moving that bar forward. We want to do better. We want to do better. So how are we going to do that? I've listed here, 30% of normal CFTR function. That's a ballpark of what we think ivacaftor has compared to a normal person. We know that has a tremendous effect clinically. Patients feel better, they do better, their lung function is better, but it's not perfect. They still have persistent disease, their sweat chlorides aren't perfectly normal, their nasal potential difference measurements are not normal. So we can still do better, we think. So how will we do that? Well, one approach is just starting earlier. And so this is being pursued, obviously, in the Kiwi study that I mentioned, by giving these drugs to patients who will benefit earlier in life. So getting in before the disease gets a chance to have a firm foothold, and before the bronchiectasis and damage has set in. Another useful approach is to treat both mutations. So ivacaftor was useful for the G551D mutation, but what if we can treat not only that, but also the other one, the delta F? And so this is being explored and tested in the VX661 study, where patients have one copy of delta F, one copy of G551D, and they get combination therapy. So the hypothesis is that ivacaftor, by treating G551D, will get a certain amount of benefit, but by now treating the delta F with a combination approach, we're going to get even more approach. We don't know the answer yet, but the studies have been done. We're just waiting on the data, so we should know soon. Then, of course, as we've mentioned, we just need more drugs, better drugs. And this is really an exploding area. This is no longer one company or just the CF Foundation. This is now multiple companies working this area, all racing to find drugs that are better and better. And so there's no doubt in my mind that in the near future we're going to have, I know we have more drugs to test, and I'm very hopeful that we're going to have better drugs available to patients. And so how do we move forward from here? I think the first thing is finding more drugs, but also continuing to refine and increase the number of endpoints that we have at our disposal to rapidly screen through and test drugs so that we can move them to patients as quickly as possible. And there's, again, no doubt we've made tremendous progress. This is by far the most exciting time in cystic fibrosis research, in my opinion, and the progress is incredible, and that it's going to continue to move in this direction until we have better therapies. And I think many of us believe that a cure is truly in near sight, that we're going to see it soon. So thank you very much for coming, for participating.
We have about 5 minutes for questions. If you could please stand with the microphone so people on the web can also hear the question.
So it's either a good sign or a bad sign when no one questions. I'm not sure which.
Hi, I had a few questions. One was, I'm a CF carrier, which we found out in the 80s when we were part of the study to find out what the markers were. And does that mean that I have 50% CFTR activity?
We think that's true, yes.
And so, because you said, and I don't have any symptoms that I know of, and so you were saying that 30% might be perfectly fine, and so carriers, if they only have 50%, that appears to be fine. So
that's right. It's actually, we had a discussion yesterday with some folks. Is 50% give you zero risk for anything? It certainly doesn't cause cystic fibrosis. I think it's up in the air whether having 50% of normal activity might predispose to something on a rare occasion. We don't, I think, know the answer to that. Mm-hmm. But certainly if we can reach 50%, we think patients are not going to have cystic fibrosis. And that even 30%, which is, again, a ballpark, debatable, but the ballpark of what we think we get with ivacaftor and G551D, that's a huge benefit. And so if we can get between 30 and 50, we're doing great.
Okay, another question I had was for the rectal biopsies or whatever you called that. Are those cell lines something that can be maintained? Like, I think I've heard like breast cancer cells are maintained and shared among researchers and things like that. And
right.
So that, because people would, I'm sure, would want individual knowledge about their own particular situation, but there also would be some benefit just to share those cell lines with other researchers.
Right. So these aren't formally cell lines, so they're not immortalized so that they can be passaged forever.
Oh, okay.
They can be passaged and expanded, which is a great thing. But I think but we know that that they can be shared, and so this is something that actually the CF Foundation and individual researchers are working on right now. And so we know that we can take a rectal biopsy in Chapel Hill and we can mail it to Cincinnati, and they can mount it in a chamber and study CFTR. So we have pretty high confidence that we can do the same thing, take a biopsy, and then grow the organoids wherever the organoids want to be studied and grown. So I think that's not going to be a big problem to have a patient with a rare mutation in Palo Alto or wherever and get a biopsy rather than sending the patient to somewhere, take the biopsy, send it, and then set up the assay. So I think that's going to be very doable.
And then my last question was, you said bicarbonate levels were important in the gut, or pH was important and bicarbonate could play a role. Sodium bicarbonate is, baking soda. Do some enzyme companies put some sort of baking soda in their pills, or would that be a simple tweak?
You're a smart guy. One company does do that. It's complicated though, and so the rationale is exactly that. If you normalize the pH, the enzymes might work better. One company adds it to their pills. A common approach done by clinicians everywhere is to use antacid type medications and again raise the pH to make the enzymes work. So it's a very common approach. So good thought.
Thank you.
Thank you very much.
Thank you, Scott. That was a fantastic presentation. Now, as I mentioned early on, we gave infections and infection control very serious consideration. So I'm very pleased to introduce to you Tracy Stoll, who is going to help us understand what we mean by infection control and what we're doing about infection control. Yeah, and we need to do a quick switch of...
Which password is it to get on?
When you're done doing this, you need to make this side of the room as dark as this side of the room.
Okay.